My name is Jamie Williamson, and I will be the host for the front row this afternoon. I'm a professor here at Scripps Research Institute. Today we have a virtual format, hybrid with people in person, so it's great. Looks like we got about 150 or more people here in person. We've got hundreds that have tuned in virtually, so welcome to everyone. Now, today the subject is going to be about brain plasticity and learning, and our presenter is Holly Cline. Now, as you all know, that when we grow up, our brains are developing. There's nothing I just spent a lot of time with some grandchildren, and their brains are still developing, okay? So I can attest to that. So as we grow, our brains make new connections, and one of the things that's very clear is that the things that you experience actually modulate what happens during this process of brain development. And then, when we experience something, that's manifested in the brain by additional connections between the hundreds of millions of neurons that are in the brain. And your brain always retains some capacity to rewire itself to assimilate new information and carry out new behaviors. And we call this plasticity. So you're going to be hearing a lot about plasticity today. Now, one of the bad news for many of us in the audience is that your plasticity declines with age. And this is a normal thing. There's probably things you can do to mitigate that. But loss of plasticity is also associated with a number of key neurodegenerative diseases and neurodevelopmental disorders. So Holly's research, in part, is trying to ask the question of how your experience in something affects how the brain develops. And so there's always this hope at the end of the basic research tunnel that we can learn something new that we can translate into medicines that overcome diseases. Now, before I'll say just a couple things about Holly, but at the end of her lecture, I'm going to come back up here and we're going to do a little Q&A. And you all can participate. For those of you who are virtual, you can type a question in at any time during the chat, and I get these relayed to me on an iPad, and I can bring them up down in front. And then if you're in the room, you can stick your hand up, and we actually have Jedi in training that have a little lightsaber and microphone, and they'll come around and you can ask your question. So I will have some questions for Holly as well. So we'll have a group discussion. I encourage you to participate. One of the things that I always like to say is there's no such thing as a dumb question, that we want you to be engaged, and if you're engaged, you're naturally going to have this dialogue with yourself. Well, what about this? What about that? So please participate in the discussion. Now I'm going to let Holly say a little bit more about her training experience, but she is the chairman of the Neuroscience department here at Scripps Research. And it's my pleasure to welcome her to the stage to talk about brain plasticity. Got all your choice? Yeah. Okay.
Thank you. Hello, good afternoon. Thanks, Jamie. I thought you were well enough equipped to give the whole lecture there. So I'm Holly Cline, as Jamie said. I'm going to tell you about brain development and the changes that our brains go through during brain development as a result of the experiences that we receive. So first, let's talk about what changes in our brain as we grow up. And one of the major questions that people discuss at this point is whether it's nature or nurture. So nature is what's in your genes, and nurture is basically what experiences you have while you're growing up. Which one of these types of influences is more predominant in shaping how our brains connect and how we behave as we grow? So You might say, well, you're tall because of your father, or you're smart because of your mom. Or you could say that you're smart because your parents read to you when you were a kid. And so these would be the different kinds of influences that either nature or nurture could impact as you grow up. So what I hope to convince you is that it's not a nature or nurture, that these two types of mechanisms or influences on your brain development interact continually, and that nature is under the control of the experiences that you have. Basically, experiences that you go through during your childhood trigger genes to become active and generate proteins that influence your brain development. So Jamie suggested that I tell a little bit about myself. So I'd just like to tell one story about my PhD advisor, Gunther Stent. So I went to graduate school at UC Berkeley, and as my thesis project, I worked on the development of the brain in a species of worm. And we did these experiments because we felt that this experimental system was tractable and that we would be able to learn fundamental aspects about brain development that would be applicable to a broad range of species. But I noticed that Gunther always seemed to be interested in something else. And what he was interested in was work that was going on at the time about visual system development. And the exciting work was all about the influence of sensory experience, of visual experience, on the development of the connections within the visual system. And Gunther made a conceptual contribution to this work by drawing a link between previous experiments that had been done by Donald Hebb on learning and memory and the work that was being done on visual system development. And he suggested that maybe there were common elements that were at play in the changes that undergo in the brain during learning and memory and during brain development. Now, the reason why this was unusual was because at the time, the prevailing view was that once the brain develops, once you're an adult, the function of the brain is completely separate mechanistically from what happens during brain development. And so to see that there might be common mechanisms underlying these two types of phenomena was a very interesting and profound consideration. But I have to say that my reaction was, what I learned from this was that if you're really interested in something, that's what you should be studying. And so I learned a lot about visual system development with discussions with Gunther, and as a result of that, I went to work on that myself as a postdoctoral student in Martha Constantine Patton's lab. And she was really an amazing, creative, remarkable person who was working in this field, and I really benefited by learning a lot from her. So that was the start of the path I'll tell you about today. So first I'm going to just give you a little bit of brain basics. Jamie mentioned this a little bit. We have about 1 billion neurons in our brain. We also have about 1 billion other kinds of cells that I'm simply calling non-neuronal cells. They matter, they're very important, but that's not what we're going to be talking about today. Neurons each form about 1,000 connections with other neurons. And these connections, these end up building networks of neurons that have certain functions and do jobs for us in our brain. Okay, so let's just take a look at what this might mean if we're trying to study these neurons and how they form connections and what rules they may use in order to form these connections. So this is, let's see, can you turn down the lights in the front? Is that possible? So this is an example of several images of neurons. They're all different colors, and there might be about 50 neurons here in this image here. That's not very many compared to the billion that you have. So let's add some more. So now we have more neurons, and each of them have synaptic connections, and they're talking to other neurons. Let's take a look at the synapses. So synapses, as shown here, has two parts. One is the part that sends information, and the other is the part that receives information. Now, this is just a little cartoon. Each different color is a different bit of a neuron. These bulbs are the receiving part of the synapses. And so imagine all of this spaghetti and meatballs in there that are connecting and have to do so in a very organized and careful way. So how can that happen? We study this question of how that can happen and how experience affects the development of the brain by studying the visual system. And the reason why we study the visual system is because we already know a lot about it, and we know that it's very important to how our brains work. So actually, it hasn't... I show this picture because I really like thinking about what we've known in the past and what we haven't known in the past. Now, Da Vinci in this diagram, he knew that the eye sent signals into the brain via the nerve from the back of the eye into our brain. But after that, he got a little confused. He wasn't too clear about what was going on in there. Thanks to functional MRI and other imaging technologies, we know that there are about 30 different areas in our brains that represent visual information that we see. And so why so many? Each one of those different areas actually does a different job with that visual information. For instance, navigation, or visual motor coordination, like when you play tennis, or facial recognition. So establishing the circuits which mediate each of these different tasks requires a lot of trial and error and a lot of refinement in order to optimize those circuits. So how does that happen? And again, we're going to go back to this nature versus nurture question and think about how nature and nurture might interact during the development of the brain. So I mentioned earlier that there had been a lot of really exciting work going on in terms of the development of the visual system. And a lot of that was initiated by these two gentlemen, David Hubel and Torsten Wiesel. They received the Nobel Prize for a lot of the work that they did on establishing the rules of connectivity within the visual system and how experience influenced the development of that connectivity. Now, you may not guess this by looking at this picture, but these guys were actually clinicians and saw patients, and they were very interested in a problem that arose during their clinical practice. And that was that when an adult walked into the office and had a cataract, that cataract could be removed and vision would be completely restored. But if a child came into the clinic and had congenital cataract, the cataract was removed, that child did not regain vision. And they were curious about what was so special during these two periods in a person's life that they would respond so differently to the effect of occluding vision and potential for recovery of that vision. And so they did a number of experiments looking at the development of visual connections, compared to adulthood, and how those visual connections might be influenced by modulating the amount of visual activity or visual experience that occurred during development. And basically what they demonstrated, and their followers, their intellectual followers have added to this information. So now we know that there's what we call a critical period of development during which sensory input into the brain is absolutely vital for the development of connections that process that type of sensory information. And in the absence of that, those connections do not form properly. And this is why when a child has impaired vision as a result of a cataract, past this critical period of development, that vision cannot recover. So this kind of work was really very influential, and it had a number of repercussions with respect to a variety of treatments, one of which is shown here. This kid has amblyopia, or lazy eye, and the treatment that we now use is to put a patch over the stronger eye. So amblyopia is when one eye has good vision, another eye has weaker vision. And the treatment is to cover the stronger eye so that the weaker eye has to work harder and gain connections in the brain that are normally equally distributed between the two eye inputs. And after patching the stronger eye for a while, they can take away the patch. The two eyes have equal access, equal strength, and they're communicating into the brain, and this kid would be recovered. Now, there are other examples of this same principle of a critical period during development that is important for the development of connections relating to sensory input. Recently, actually just within the last month or so, there have been 2 independent reports about gene therapy for congenital deafness. These were treatments for children who were deaf from birth. They had no hearing whatsoever. They received viral gene therapy of the gene that was missing in their ears. And very shortly after the therapy, these children were able to hear sounds. But it's very interesting because it took another 6 months or maybe a year for these children to be able to understand and process those sounds and to gain language. Now, we know that language means that you have to be able to hear sound and you have to make sound and match those 2 sounds and through trial and error. And I found it very interesting that there was very clearly this relatively rapid ability to hear and a delayed response of learning to actually understand what those sounds meant and to use those sounds for language. So another example is in facial recognition. And there's been a long, long debate about whether facial recognition is innate or acquired. And Marge Livingston, who worked in the labs of David Hubel and Torsten Wiesel, have spent many, many years trying to sort this out. You can imagine it's quite a challenging experiment. And she finally brought together enough data to convince both herself and her critical colleagues that indeed facial recognition is an acquired feature of our brains. It requires that same exposure and that same sensory practice as other types of sensory input. Okay, so how do these brain circuits get hooked up, and what does that have to do with nature versus nurture? So, when I was a kid, we lived in New York, and there was a lot of Yankees games that we listened to and watched. And Yogi Berra was a Yankee. And he had a lot of funny sayings, one of which is shown here, that you can see a lot just by looking. And when I started my independent lab, I wanted to be able to actually see in real life how neurons were growing in the brain, in the visual system, and how they might be influenced by visual activity. And so I was very lucky because Scott Fraser, who was at UC Irvine at the time, and his postdoc Nancy O'Rourke had just established the methods of labeling individual neurons in the brains of these transparent frog tadpoles with fluorescent dyes, and then he could put them onto the stage of a very relatively new microscope, a confocal laser scanning microscope, and collect time-lapse images of the same neurons and watch them grow in the intact living animal. So I went and learned how to do this from Scott. And this is an example of one neuron that I thought was very beautiful. We collected a lot of time-lapse images of neurons as they grew. And this is an example of one image series. So this is one cell that we imaged in the intact animal, initially at 2-hour intervals over 6 hours, and then once a day over about 4 days. Now you can see by looking at the first image and the last image that the arbor, the tree part of this neuron, which receives information from the eye, grew over this 4-day period. We were interested in what was happening here. So when you could see that there weren't that many changes, maybe just a few little dancing bits and pieces in the arbor, but no huge growth. But we were wondering whether or not those little minute kinds of modifications might be important for the net increase, the net elaboration of the arbor over time. And so we looked more. We looked a lot at these cells. Here you see this neuron. Every little bit is dancing, extending and retracting. And the reason why we thought that was important in terms of how vision might influence the way that these neurons were growing was because of this experiment. What we did here was take an individual neuron and looked at it using a different microscopic technique called electron microscopy. And this allowed us to totally reconstruct the arbor of the cell and identify where every single synapse was located on that cell. And what you can see here is that every little branch has a synapse on it. But I just showed you that every little branch is coming and going and wiggling and extending and retracting. So that meant that a lot of these synapses had to be very dynamic, be added and retracted at a quite rapid rate. And so, in homage to Hubel and Wiesel, we carried out an experiment like this, where we wanted to introduce visual stimulation to these tadpoles and test whether or not this would modify the dance that the neuron dendritic arbor was going through. And this is an example of the kind of data that we acquired during this experiment. So again, here's the reminder that every one of these branch tips is very likely to have a synapse on it and receive information from the retina. So the experiment is to label a single neuron, again, in our transparent tadpole, put them onto the stage of the confocal microscope and collect an image through the individual neuron. Then we took the animal off the scope and put it into the dark for 4 hours and then collected another image, shown here. And then we provided the animal with that motion stimulus that I had just shown you and collected a third image. Now, I hope all of you can see that if you compare this tree and this tree, they don't look so different. There's not a lot of change going on there. But if you compare this tree and this tree, you can very easily see that there are a lot more branches and therefore a lot more chances for synaptic connection and input from the retina. So this told us, this was sort of the living example of the experiment that David Hubel and Torsten Wiesel had done, where they had shown that visual experience was very important for the development of individual neurons and connections within the visual system. So as a cartoon version, what we showed is that you start out with a younger neuron, it has some synaptic contacts, you provide visual stimulation, the neuron elaborates a much more complex dendritic arbor, and there's lots of input onto that cell. Now, the visual activity does other things as well. It also increases the assembly of the circuit, of the network that's processing that visual information. It enables visually guided behavior, so now the animal can swim away things that might want to eat it. And it also assists in recovery from injury. So in a separate set of experiments, we injured that part of the brain where these neurons are located, and we provided visual stimulation and demonstrated that that enhanced the rate at which the brain could recover from the injury. So how do all these great things happen? So you probably all heard about the central dogma. We all have DNA. That DNA is transcribed into messenger RNA. And from this messenger RNA, we get proteins. Proteins are translated or synthesized from the mRNA template. So at about the time just after Hubel and Wiesel had made their discoveries about the influence of vision on brain development, there were other studies which indicated that active brain cells can turn on special genes, which we called candidate plasticity genes. And two people who carried out these types of experiments are Ellie Nadivi and Paul Worley. They were working independently. Ellie was at the Weizmann Institute. Paul Worley was at the Johns Hopkins Institute. And basically what they both did was do experiments where they activated the brains of animals and they isolated the messenger RNA to see if there was more and different kinds of mRNA that was present in the cells as a result of the brain activity. And what they discovered was that there were hundreds of these candidate plasticity genes. And they subsequently tested these genes and discovered the roles that each one of them plays in brain development and plasticity. And I'll just show you this one example. I was very lucky that Ellie came to work with me in my lab. She had all this expertise in molecular biology, and I had a lot of expertise in imaging, so it was a great combination, and she did this kind of experiment represented here. She took candidate plasticity gene number 15, and she expressed it in the individual neurons within the brains of the animals that we could image over time. And you can see, compared to neurons that did not receive that gene, that the ones that received the candidate plasticity gene had bigger and bushier arbors than the neurons from the control untreated animals. And what this means, I'm not going to show you the data for it, but what it means is that they have more synaptic connections and stronger synaptic connections. So all the things that a tadpole's visual system is supposed to do for them, this guy could do it bigger and better. So we think that this same kind of effect of brain activity is operating in each of these conditions as well. So, okay, we get DNA to mRNA. Those are the genes that Ellie and Paul Worley both discovered and studied. And then you get proteins. So we wanted to find out what proteins are synthesized in response to brain activity. So why do you want to study the proteins? Well, it's not always true that when you get DNA to RNA that you get exactly the same proteins being made. There's not a one-to-one correspondence there. So that's one reason. Another reason is that proteins are what actually do the work in the cell. It's not the mRNA. The proteins are what builds our cells and how the cells function. And the other reason, as Jamie said, ultimately we're all hoping that maybe somehow there will be therapies that come out of some of the experiments that we do. and proteins are usually the targets of drugs. So we were very curious to find out what proteins are synthesized in response to brain activity. And this was right about the time when I moved to Scripps, and that was really important because there is so much phenomenal expertise here at Scripps in terms of being able to carry out these types of experiments. And so I had a remarkable graduate student, Han-Chen Liu, and a postdoc, Lucio Chiaparelli, who worked very closely with John Yates here at Scripps Research Institute and his postdoc staff scientist, Dan McClatchy, to help us do these experiments. So what we were interested in doing was to screen for newly synthesized proteins that were generated as a result of visual experience. And so this took quite a variety, a combination of new technologies that we assembled with John Yates' assistance. And what we found was that about 25% of the proteins that were newly synthesized in response to visual stimulation were actually generated from genes that were autism spectrum disorder risk genes. And that was unexpected to me, because I think typically autism is not thought of as a disease of brain activity. And so this, again, I thought it was quite interesting to think about. And then the other thing is that many of these genes were actually FMRP targets, and I'll talk about that in a minute. Okay, so what this indicated was that visual experience does increase the expression and synthesis of candidate plasticity proteins. And now we have a chance to dig a little bit more deeply into what those are. But I mentioned FMRP. So FMRP is an interesting protein. This is the protein that is not functioning correctly in people with Fragile X syndrome. And what FMRP does is it binds to mRNAs and sequesters them. It's sort of like it takes the standard DNA, mRNA to protein, and it puts a weight box in there where the RNA are sequestered by this protein, this FMRP protein, and they can't be synthesized into proteins. And so what this experiment that we did indicates is that while FMRP is harboring those mRNAs so that they're not able to be translated, somehow visual experience affects FMRP function so that now those mRNAs are free to be translated into proteins. So again, this wasn't anything that I was aware of with respect to autism diseases or FMRP, or Fragile X in particular. So that was exciting, and then we wanted to do more, and we wanted to do it bigger and better. And so this was our version 2.0. We were interested now in screening for changes or increases in protein synthesis in response to visual stimulation in a cell type specific manner, because there are lots of different neuron types within the brain. So we wanted to be able to get higher resolution, which types of cells are changing in which types of ways. And we also were interested in looking at different times in development compared to adulthood. This work was done by Yi, and she's sitting here in the audience with Roxy Wang, who is a technician in the lab. And what they did was took the pipeline that we had used previously and improve it in every way possible. So we optimized the pipeline in terms of the timing, we improved our technology, and we got a much higher yield of newly synthesized proteins than we had been able to do before. So that was great. And I'll just tell you one highlight. So what we found was that our top, number one, most highly expressed candidate plasticity protein was probably one of these mRNAs that was held into a box and could not be readily synthesized until there was a signal from the visual stimulation, which then resulted in a huge increase in this protein within the neurons. There were also a lot of many other candidate plasticity proteins that were synthesized in the more traditional way. But the other interesting thing about our number one candidate plasticity protein was it very rapidly ended up putting a block on further synthesis of other proteins. And so I find this very interesting because it means that there's a mechanism where activity can trigger protein synthesis. A whole bunch of new proteins are synthesized, at least one of which now has the responsibility of putting a block on any further synthesis. And what this allows the cell to do is have temporal control over the flood of new proteins in the cell, which can be then used in a controlled way to build new connections and to refine the connections that are triggered as a result of the visual experience. So this CPP candidate plasticity protein number 1, it also does a few other things. Yi found that it regulates synapse formation and maturation. She found that it's required for vision. If you take it away from the visual part of the brain, it's as if the animal can't see well anymore. And it's also required for depth perception. And that's, for instance, one way that we can test how is the animal using this information? How is the test for depth perception? So this protein seems to be activity regulated and to have a very broad impact on the development of visual circuitry. So I'll close there and just say full circle that your experiences, what you receive as nurture, as we call it, activates your genes, nature, and this then contributes to the machinery, the building machinery, and what fine-tunes your brain and your circuits. Thank you. I just need to take one more second to thank a few people. So this group was my group in Cold Spring Harbor before I came to Scripps, and they were really wonderful, and we discovered a lot of exciting things. This is the group here that has been with me here at Scripps. Again, a fantastic group of people to work with, and I've enjoyed every single one of them and all the excitement that we've had in the lab. I'd particularly like to acknowledge Helen Doris and her generosity and support for the Doris Neuroscience Center. I have wonderful colleagues in the Doris Neuroscience Center, and we all enjoy our interactions and trying to do the best science that we can. Of course, this wouldn't be possible without financial support, so I've received continuous support from the National Institutes of Health, and I particularly appreciate the NIH Pioneer Award, which I received, and with it, I said I just took the jump and started to do all the protein work that I told you about most recently. And of course, our colleague, John Yates, and the people in his lab who have helped us out. Thanks again.
Thanks, Holly. You know, it's something I've done a million times, but you're like in the heat of summation, and you forget the most important slide of all, which is the great people that do all of this. I've done it, and so I'm glad you got to that. Again, if you have a question, put your hand up. I have a couple things. So CpG 15. This was your protein that grew in the tadpole, that grew more arbors when you shined light on it. So that's what happens to the neuron. So is there anything about that animal that you can tell that it saw light? So is there something in its behavior in response to light that you can tell that it grew some more synapses and grew some more arbors and made more synapses?
Yeah, so one additional interesting thing, so the CpG15 was expressed in the cells I showed you. The cells that sent axons and made connections with the cell in the picture also got bigger. So there was a signal that says, okay, let's form strong synapses. And then another part of the conversation was to increase the input. So it was like it amped up the whole circuit. It was really amazing. And we didn't do any, that's the nice thing about working with animals that have more complex behaviors. We can ask whether or not it affects their depth perception or something like that. We had trouble figuring out how to ask a tadpole if it sees better.
Okay, all right, understood.
Yeah.
So one of the things I just kept thinking about through your whole talk was, to zeroth order, we aren't really growing a lot of new neurons in our brain. Or maybe that's not true.
But, well, even this very moment, you're growing new neurons in your hippocampus.
Okay, all right. So, but we talked a little bit about declining and not being able to learn new things, but as adults, you can learn things. Mm-hmm. And so what's happening? Is it that you're retracting some synapses and exploring new ones? And then maybe say a little bit more, because I think one of the obvious things is that when people have traumatic brain injuries of certain kinds, they can recover function. And it must be what you're studying underlies that very phenomenon. So,
yes, so that was part of what I thought was exciting, to sort of draw this connection between the events that occurred during development. And even though we have less plasticity as adults, it's still a lot of the same stuff that's changing. New synapses are made, in some cases, new neurons are born. And so it's still, in my opinion, it's still a little bit confusing, because if we are able to learn stuff, that's clear. But what about all those things we learned before? Do we really only have limited amount of information capacity? Or how do we condense information and store it more efficiently? So these things, in my opinion, are not clearly resolved. But the mechanisms that operate during brain recovery from brain injury and learning even as adults, they're very similar to what happens during development.
Well, I can tell you personally, my inbox is full. So maybe a related question, and this is from one of our online viewers, and it has to do with seniors. And so should we continue to have stimulating activities in order to help repair our neuronal circuitries?
The mantra is exercise physically and mentally, and that will, to say it'll keep you young may be a bit of an exaggeration, but it's really the elixir. It's the elixir, okay. Okay. And another question from our virtual audience, and you touched on this, and you said that some of the things you found were, associated with autism. So does neuroplasticity change over the lifespan of an autistic person in a different way than? Oh, actually, I forgot to say, with respect to autism, so there's a new treatment now for autism where, and it is specifically effective for people with Fragile X syndrome. And so what I showed you was that this Fragile X protein prevents other proteins, mRNAs, from being translated. And the new therapy that is now being used quite effectively is one that increases activity through a particular type of glutamate receptor that then influences the brain activity or the neuron activity with in those people, that increases protein synthesis. So I just wanted to say, completely separately, we're doing this super basic research, and then other people are devoted to trying to find therapies for Fragile X, and it seems like there's been convergence in terms of understanding how these things might be working together. So I don't think that was your question, but...
Yeah, that's great. That was great. We have a Jedi over here. So.
Hi, thank you so much. That was a really fascinating presentation. Just to dig into Jamie's question about how much of this neuroplasticity might be a zero-sum game, two of the examples you gave, one of lazy eye, I know I've heard anecdotal reports of people who've had that treatment where the strong eye has been patched, and then after the treatment, they feel as if that strong eye has actually become weaker, at least in comparison. And I know with the latest gene therapies for congenital hearing loss, there have been reports from these children of potential overstimulation in the months immediately following the treatment. And I'm wondering, how much do you think that that might impair their previous skill of maybe sign language that they've developed? And how much do you think that potential overstimulation or new stimulation might interfere with old skills? And how much of this neuroplasticity needs to balance out acquisition of new skills with maintaining old ones?
Yeah, I think, thank you for that question. It's very interesting. It's what that means to me, those two examples that you mentioned, are that if we're trying to correct these errors of sensory input, either through deafness or through lazy eye amblyopia, it means that we're trying to reestablish, for instance, the amblyopia is easy to think about because it's a balance of trying to gain comparable input from both eyes. And the magnitude of the strength of each of those inputs is driven by the strength of stimulation. However, if one eye sort of gets used to working with a certain level of input and then changes dramatically, you can imagine that there might be some kind of adjustment that needs to be made in terms of those contacts in order to get exactly the 50-50 that we're imagining that would be ideal. And similarly, I think in terms of these kids who get the gene therapy for congenital deafness, imagine where you didn't have any auditory input and then all of a sudden there's just all this racket in your brain. I mean, I can totally appreciate how we, or physicians working with the patients, would have to be very careful in modulating the amount of sensory input so that the brain can actually deal with it, as opposed to having some kind of mechanism that might actually shut it down.
Okay, I have another virtual question, and we touched on this a little bit, but obviously when the brain is developing, there's this kind of balance, like there's the right amount of connections, and that number of connections, as you showed, depends on stimulation of the appropriate kind. So what happens, and are there diseases that are specifically associated with not having enough or having too many of these connections? So hyperproliferation of synapses.
Well, there are phenomena where there's hypersensitivity, for instance, to touch. That's quite easy to have somebody report on, right? But you know, another interesting thing about the brain is this sort of magic stuff called homeostasis. And homeostasis is a phenomenon where you always try to return back to a center ground, right? So you learn a lot, maybe you forget over here, you only have so much capacity for a certain capacity for a certain number of synapses, or synapses can only be so strong before they start to weaken. And it really, if you don't understand something, you can just say, oh, that has to do with homeostasis. And yeah, it's really complicated. I mean, and I'm saying that, you know, seriously, because we describe this phenomenon, but we really don't know how it happens or who gets to set the middle ground, right?
Yep. Yeah. Okay. Another Jedi over here.
Hi, I was curious about the redundancy in the system, the plasticity gene 15, which we are talking about, how important that is. And suppose it was to be knocked out, and then what kind of impact would it have? And it can be done in experimental situations where one can knock out a particular gene and see what's the impact of that. Does that result in now a compensating gene being activated, which now produces another protein which takes over the function? Or you lose a function entirely of regulating the MFR, that box which you said is there. So what's the level of importance of having this gene available and the consequent protein which is there?
Yeah, the way that we attempted to decrease the function of CpG15 was by expressing a type of the protein that we thought would be less functional. That was, and those cells had less connections and less elaborate dendritic arbors. And but they weren't like totally shrimpy. It's not like CpG15 was the only gene that was responsible for the elaboration of the dendritic arbor. There's many different components which contribute to that outcome. And so I would say that it's very rare that you remove one component and it's so fundamentally vital, or another way of thinking of it, it's so upstream in the set of processes which occur during development that it would result in, for instance, a neuron dying. But that does happen. It just didn't happen with CpG15.
Oh, we have one. Oh, one over here. Yep. Great.
Okay, first I'm going to start with, like, I couldn't do math and I couldn't do science, so I had to be a lawyer. So I'm going to ask my probably incredibly stupid question. If I understand what you said, that it is really, really important to be able to see when you are young. Is that correct?
Yes.
So what we should do is make sure that we test all children for vision and get their vision corrected as soon as possible. Is that a takeaway from what you said, or did I miss the entire port?
So first off, my sister's a lawyer, so it's all fine. So I would say that it's very valuable to have children tested for all of their physiological and sensory functions, and to try to bring them up to a certain level where their brains that process those different types of sensory information will be able to form. Now, you may know, so I talked about this gene therapy for profound deafness, for congenital deafness. The deaf community is not totally excited about this, because they feel like, and I just heard an interview with someone who wished that all of this money and effort that had gone into the gene therapy for deafness had been spent on something that would have, the way this person said it was, prevented children from dying. So they just feel like people who are deaf, they're just fine. They're just not like other people who can hear. And so that's why I want to qualify my statement. Personally, I feel like, yes, everybody should have all the sensory modalities that we have available to us. That's how we enjoy our lives. But Other people would not agree with that.
I guess what I was thinking about was, let's look at the children who are economically deprived and focus on making sure that they have their vision tested to give them the most opportunity.
Absolutely. Yes, absolutely.
I think we have a question down here. Front row.
First of all, thank you. Your presentation was wonderful. Some of it I actually understood. I am aware of myriads of experiments with gene therapy in all sorts of areas. And yours is extremely interesting. Do you ever feed all these wonderful experiments you've done and all the outcomes that you've noticed and conclusions you've drawn into a giant computer and run it against all these other experiments to see if two different therapies or many different therapies can be combined as breakthroughs?
Yeah, that's a great question. So one transformation that's occurred in the biomedical research field is that now the data that many of us collect is available much more easily, much more readily for other scientists and other clinicians to have access to. And so you can actually say, well, I have this idea, I've made this finding. Let's see if anybody else made this finding or if they reported something similarly. So the old-fashioned way was you open up a journal and you read it. But now there's just much more access to all the information that has been published. And I would say it has been just remarkably transformative for science.
So it bodes well for the future.
Absolutely, yeah.
Thank you.
We actually have scientists whose job it is to
Yes,
assemble and organize all this information and to make sure it's properly disseminated. That's a whole field, and the advent of genomics techniques. Holly talked about RNAs and proteins and DNA. There's an avalanche of information that's coming out, and there's some very thoughtful people organizing that information so it's readily accessible to the community. I'm astonished at what you can find. We are coming up on the hour. Okay, we'll take one more question.
I just have one question about the gene therapy. Are there stem cells involved with that as well, or nerve growth factors?
Not in the therapy for the deafness, no.
It was a viral delivery.
Yes, it was a viral gene delivery strategy.
Yeah.
We have time for
Okay, I want to...
Wait, there's one more.
Oh, wait, there is one more? Oh, sorry.
I had a question about the tadpoles. Did they have any self-awareness of the experiment? Did they seek the light?
Sorry, did they have any what?
Awareness of the experiment. Like, for instance, you were completely controlling how much stimulation they had. So there was never any time where they could opt in, opt out. I know you're laughing, but I think it relates to the whole question about, when is it beneficial and when does it become overstimulation, and is there the ability to have a sense of that?
So the experiments that we did with the tadpoles, they would live their tadpole lives, and then we would do our experiments. As you saw in the example I showed you, it was for about 8 hours out of the day, and then they went back to their tank. And
So you controlled totally the exposure.
mm-hmm.
Okay. Just a related, just as the mother of a very premature baby, when my baby was born, there was a thought that the neural connections go through a point of pruning, actually, towards the end of the gestation, just shortly before birth. And so there were issues in terms of whether preemie brains had excess connections. Is that still the thinking that...
Yeah, so actually, I didn't bring this up during the talk, but it is germane to your comment. So it's still actually in the textbooks that neurons grow really big and then they prune a lot, just like what you said. And in human development, it often happens during the 3rd trimester. But what we observed with our particular type of imaging is that there's always this trial and error, this sort of approach and avoid, kind of extend a branch, pull it back. And so it's quite different than the way that it is thought to occur. And I say that because you could, it's like, you can learn a lot more if you actually look in detail at the processes of how something changes over time. Because what you were told about your child is that the nerves would grow, and then in a healthy developing child, they would retract and sort of fine-tune. And that may also have occurred under the circumstances of your child. But that's not really how it works. There's always this sort of back and forth test kind of going on throughout development.
Okay, very good. I think we'll take a pause here. I want to remind those of you who are here in reality that there's a reception outside, and those of you who are here virtually, we would encourage you to come to campus for the next one. This has been a really great discussion. I can tell everybody's engaged, and we're really happy that you could come here and experience one of our great scientists. We have a deep bench, and so I would encourage you to come to the next front row, which is March 20th, and Professor Hugh Rosen is going to be telling us about new medications that he's developed for mental disorders. So on March 20th, you will find me in the front row. Thanks for coming.